Hello, in this video we'll be examining chromatin immunoprecipitation applications in embryo research. Chromatin immunoprecipitation is a powerful method for investigating DNA-protein interactions. Combined with next-generation sequencing, finding the exact binding sites to which the protein of interest is bound is possible. Step 1 is fixation, where proteins are glued to DNA using formaldehyde. Here we could see that all proteins are glued to the DNA with a formaldehyde solution. In step 2, it can be seen that fragmentation of DNA occurs with sonification and restriction enzymes. First, it's reported optimized fixation and sonication techniques that yield chromatin cross-linked and sheared enough to detect transcription factor occupancy at promoters with at least 1000 pp resolution. Second, because the standard 1% SDS lysis buffer used during sonication tended in our hands to produce low-quality chromatin from early embryos, we use the low SDS 0.1% radioimmunoprecipitation assay buffer. RIPA buffer produces high-quality sheared chromatin samples while reducing yolk solubilization, thus limiting background by preventing protein precipitation. Doubling the number of washes further reduces background. In step 3, immunoprecipitation occurs with the beta-catenin antibody against the beta-catenin protein. In step 4, purification to remove protein DNA interactions that are not the target interaction occurs. From the article, it is stated, finally, we have optimized DNA purification and PCR conditions to allow for the reliable detection of as little as 30 copies of a target sequence per reaction, facilitating the use of as few as 5 × 10 to the 4th cells in the starting sample, an improvement of 2 orders of magnitude from the typical precipitation protocol. In step 5, reversal of cross-linking occurs. Here, protein is separated from DNA and DNA is then isolated. This will lead to step 6 of the protocol. Here in step 6, DNA is now isolated and can be sequenced. Down below is the DNA fragment that is sequenced. Figure 3 is proof of success using this methodology to find the beta-catenin protein binding site. The specificity of the beta-catenin antiserum was confirmed by performing chemoprecipitation on blastula stage 9 embryos and competing with an excess of immunizing peptides. Lane 2 is pre-incubation of antibody with peptide, while lane 3 is the addition of peptide directly to sheared chromatin sample. Following IP, samples were processed as described in the text and separated by 8% SCS page. Immunoprecipitated beta-catenin was analyzed by a standard Western blot using the beta-catenin antiserum. Peptide competition of the beta-catenin chemoreception and in C knockdown of beta-catenin by morpholino injection confirms the specificity of the interaction between beta-catenin and the XNR6 promoter. The competition in B and C were performed by pre-incubation of the peptide with the antiserum for 1 hour before addition to the sheared chromatin sample. Thank you for watching.